Good morning. So this is a recorded presentation for the topic antigens, the characteristics of antigens. An antigen is any substance that, when introduced to the body, can stimulate the production of an antibody. So it originated from the notion that it can stimulate antibody generation. So that's why they use the word antigen. There is another related word which is called immunogen. So this is when an antigen can induce a specific immune response. Okay, they are almost similar. Another term is epitope or antigen determinant site. So this is the part of the antigen that combines with the products of a specific immune response. Okay, so when an antigen is supposed to induce a response, and there is actually unresponsiveness, we call that tolerogen. Allergen is when an antigen can induce anaphylaxis or a severe type of hypersensitivity reaction, right? So something like medicine or pollen grains, or even seafood. So that is allergy, some kind of allergic reaction. Tumor antigens are those that are found in the MHC molecules, one molecules on the surface of tumor cells, and they can sometimes be presented only by tumor cells and never by normal cells. In this case, they are called tumor-specific antigens, and they typically result from tumor-specific mutation. Okay, so the next term is called an autoantigen. That is when a protein or complex of proteins, DNA or RNA, is recognized by the system of patients to be um, something foreign. So that will lead to a specific immune response. Vaccines are antigen preparations that can induce protective immune response against microorganisms and are usually done to prevent diseases. So there are many types of vaccines. For example, killed vaccine, okay, specifically rubella virus, attenuated or altered vaccine, measles, and toxoids from tetanus toxin. An antibody, on the other hand, is something like a protein that is produced to respond to an immunogen and it reacts with an antigen. So we are now going to compare the two terms that we have encountered earlier. Immunogenicity is the ability of the molecule, the antigen, to be recognized by the host immune cells and elicit an immune response. So this immune response is not necessarily production of antibody, but if the immune response is to produce antibody, okay, or lymphocytes, then it's called antigenicity. Not all antigens are immunogens, and all immunogens are antigens. Antigens can be classified depending on the following. They can be complete or incomplete. Okay, they can be classified according to their origin or source. Okay, whether they need the help of T cells or B cells to produce antibody, and based on their chemical. An incomplete antigen is called a hapten. So this is a substance that is non-immunogenic, but it can react with the products of specific immune response. They are small molecules that could never induce an immune response when administered to a carrier, even when you have a carrier molecule. These free haptens can react with two products of the immune response, and after which these products have been elicited. Haptens have the property of being antigenic, but they are not immunogenic. What about a complete antigen? So they are proteins or somehow related to proteins in terms of structure with large molecular weight, and capable of stimulating the immune response by themselves. Majority of human pathogens or microbes and their toxins are classified as complete antigens or immunogens. Exogenous antigens are those that come from outside. So they may be acquired through inhalation, ingestion, or injection. So that is the mode of transmission, and by endocytosis or phagocytosis. These antigens can be taken into antigen-presenting cells or APCs and processed into fragments. Endogenous antigens, on the other hand, are those that have been generated within the cell. Okay, this is because of normal cell metabolism or because of the presence of virus or intracellular bacterial infection. Right, so examples of these would be the autoantigens that may be protein or even a complex of proteins like antigen-antibody complex, given the nucleic acids that can be detected by the body as an antigen. Tumor antigens are those antigens that, again, presented by type 1 MHC molecule on the surface of tumor cells. So they are classified as endogenous antigens. Another classification is when we look at the chemical composition of the antigen, they may be made of proteins, they may be made of polysaccharides. So it may be a combination of proteins plus carbohydrates or fats or lipids and carbohydrates. But okay, of course, if they are made of a protein or combination of protein and another substance, they are considered as very good immunogens. Polysaccharide antigens are made up of polysaccharides, so that is carbohydrates and lipopolysaccharides, and are also considered as good immunogens. Nucleic acid antigens are poorly immunogenic, but they can be immunogenic also when they are single-stranded or when they are in complex with proteins. Lipid antigens are usually non-immunogenic and somehow they can act as haptens. And some glycolipids and phospholipids can also stimulate T cells and produce a cell-mediated immune response. Right, so there are also antigens that they need the presence of T cells in order to produce antibody. Examples of these would be the protein type of antigens. They are characterized by a few copies of many, having many different antigenic determinant sites. So an example of this would be the microbial proteins and proteins that are altered or non-self. The other one would be those that are independent of antigens. They can directly stimulate the B cells to produce antibody without the requirement for a help from the T cells. Right, so the production of antibody is almost exclusively IgM in nature, and there is no secondary immune response. Okay, so superantigens are those that can cause the T lymphocytes to um, simultaneously bind with class II molecules and at the same time act as mitogens. So that means they can undergo mitosis. They can stimulate the production of polyclonal T cell response. Comparing that to the conventional antigen, which is the traditional or the common antigen response, and that is they produce monoclonal or oligoclonal T cells. Examples of these would be the substances produced by staph and strep. They are the toxins produced by these organisms, gram-positive organisms like enterotoxin, toxic shock toxin, exfoliating toxin, and pyrogenic exotoxins. Okay, so in this diagram, you can readily see a comparison of conventional antigen and a super antigen. You can see there that in a conventional antigen, there is a need to identify the part of the antigen which is called the epitope But in the case of a super antigen, they can directly stimulate this uh, class class 2 MHC molecule to trigger a response. Right, so this is another way to show how super antigens work. Right, so they have this direct stimulation of the T cells, okay, by binding with the class 2 molecules. There are certain factors that can affect immunogenicity of substances. Okay, and these factors could be related to the antigen itself, or it can be related to the host or the biologic system. Okay, so the factors that are related to the antigen itself are number one, foreignness. So the immune system usually discriminates between self and non-self, and only foreign molecules are considered immunogenic. Another factor is the size. So immunogenic substances are generally larger. Okay, and how large should it be? In this case, it's saying that the size should be about 50 to 10 times 10 to the third Daltons in terms of molecular weight. Okay, they can also be a little smaller than that, or even smaller than 10 times 10 to the third Daltons. In terms of chemical composition, so we know that certain antigens are proteins made up of proteins or in combination with other substances like carbohydrates or lipids. Right, so complex proteins are considered potent immunogens. Another factor is the physical form. That is, if they are particulate, they are more antigenic or immunogenic compared to those which are soluble. And another thing is when they are denatured, they are more immunogenic compared to the native structure. Next is degradability. So this is something to do with antigens being able to be engulfed by macrophages or phagocytes. It is because it's necessary for processing of those antigens before they are presented by these antigen presenting cells. As for the factors that are related to the host, the first one is the genetic factor. So one is the individual may actually have a lack of genes that code for the receptors for antigen on the B cells or the C cells, okay, or they may not have the appropriate genes that are necessary for the antigen presenting cells to present the antigen into the helper cell. So in this case, it becomes a problem. Number two, age. So it can also influence the immunogenicity. So the very young and the very old have diminished ability to mount an immune response compared to other people. Another factor is how the antigen is administered. Number one under this would be the dose. How much of the antigen was administered? Of course, there is a necessary amount for the immune system to be triggered. And then number two is the route of administration. So generally, subcutaneous route is better than IV or in the gastric route. Okay, and then adjuvants. So these are substances that can enhance immune response to an immunogen. Okay, so our immune system can actually recognize certain molecules, highly conserved sets of molecules, because of the following. So we have the PAMP and we have the PRR. So PAMP is short for pathogen-associated molecular patterns, and PRR is the pattern recognition receptors. So the first one, PAMP, are molecules on the surface of the microbes, and it is capable of being recognized by the innate molecules, immune molecules. The PRR, or the pattern recognition receptors, on the other hand, are molecules that are found in the cells or molecules of the immune system, and they are again capable of recognizing um, foreign substances. So these molecular structures are specific to the microbes, the different organisms, because of certain germline receptors called PRR. The, there are several types of PRRs, and they can perform a specific task like opsonization, activation of complement, and phagocytosis, and more. The innate immune system can recognize this in three ways. So the PRR can recognize the microbial components known as PMP, and this is essential for the survival of the microorganisms. The second method is that PRRs are expressed constitutively in the host and can detect the pathogens regardless of the stage of life cycle. And thirdly, they are also expressed on the cells of a given type and independent of immunologic memory. Different PRRs react with specific PMP, and when they do, they have different patterns of expressions. They can activate certain signaling pathways, and this will lead to anti-pathogen responses. So the basic machineries that underlie this immune pattern recognition is highly conserved among species, even from plants or fruit flies to mammals. Okay, there is what we call TLRs, or the toll-like receptors, and they have the ability to recognize pathogens or even derived products from the pathogens, and they can initiate a signaling event that lead to the activation of the innate host. TLRs, or toll-like receptors, are, as we mentioned, pattern recognition receptors, PRR also, that are activated by some parts of the microbe, some part of the organism, and also some part of the molecules of the host. They signal by initiating acute inflammatory response by induction of the antimicrobial genes and inflammatory cytokines or chemokines. So those are the ways by which TLRs are able to initiate these processes. Subsequent events can happen, like, for example, recruitment of neutrophils, so that is because of chemokines, activation of macrophages, so that is because of the presence of antigens, okay, and it can also lead to the killing of the microbes. Okay, the TLRs are the primary sensors by which we can recognize the pathogens, and they are, of course, responsible for initiating, orchestrating the responses of the innate immune system. They contribute significantly to the activation of adaptive immune response. So they are also related to adaptive immune response, and there are about 10 TLRs. They are named like TLR1 up to TLR10. The receptors that are present can recognize the molecules derived from different microbial agents, right? So, okay, in this table, we can see the PAMP, the PRR, and what will happen when they are activated. So the first one that can be detected would be the cell wall of the microbes or bacteria. And the PRR that is responsible for the detection of this would be the complement, and it leads to opsonization and complement activation. Another one would be the mannose, which is also present in the cell membrane of the organism. Okay, this can be detected by the mannose binding protein, and again, it can lead to the same biological reaction. The third one here would be the polyanions, which is detected by scavenger receptors, and it can lead to phagocytosis. And the last one in this table would be the lipoproteins that are present in gram-positive bacteria, and even the cell wall of the yeast, okay, and the components of these substances. So TLR number 2 is responsible for its detection, and it can lead to activation of the macrophages and secretion of certain substances that have inflammatory effect. Okay, continuing the table, double-stranded RNAs can be detected by TLR number 3, and this can lead to production of interferon because this is found usually in viruses. Lipopolysaccharide, which is found in gram-negative bacteria, is detected by TLR4. It can lead to activation of the macrophages and secretion of cytokines that can cause inflammation. Flagellin for flagellated organisms, TLR5, so again it can lead to macrophage activation, and secretion of substances that can lead to inflammation. TLR7 can detect uh, single-stranded viral RNA, and this can lead to production of interferon, CpG containing the DNA. This can be detected by TLR number 9, and it can cause again activation of the macrophage and produce cytokines that can trigger inflammation. Okay, so the host cell can have many different toll-like receptors on the surface, and each of these can detect different parts of the organism. Right, so the reason why they call these toll-like receptors is like if you are in a expressway, in the expressway there is what we call a toll gate. So supposedly each gate is capable of allowing certain types of vehicle to pass through. Example, there is a toll gate designated only for trucks. There is a toll gate, there's a responsible for accommodating other vehicles. So it's also the same here. So it's also showing the possible effects of interaction between the PAMP. Okay, this is the PAMP and the toll-like receptors, or the PAMP, PRR rather. Okay, so in this diagram, we can see that the different receptors, the toll-like receptors or PRRs, can detect different parts of the microorganism. It can be the lipopolysaccharide, if it is a gram-negative bacteria, or it can be like in a PRR, it can detect other substances. So there is recognition in short. So there will be a response. What will happen when this is there is recognition? So there can be, because this is innate immune response, so it is more like cell-mediated immune response. So there will be phagocytosis or production of production of certain substances, the cytokines that can lead to these outcomes. What will happen? So if it's the release of chemokines, it can lead to invitation of neutrophils to go to certain tissues with the microbes. It can lead to direct killing of the microbes also. Okay, so we can see here the different TLRs, different TLRs, and what they are supposed to detect. Okay. One of the topics that we have been talking about in previous sessions would be antigen presentation. So first, the antigen must be processed, and this involves the interaction of the PAMP and the PRR, followed by the digestion of the foreign substance by the phagocytic cells. So antigen presentation is when the peptide antigens is shown by the antigen presenting cell or the APC, and this is associated with the MHC molecules, and it will be presented to a T cell. The path that leads to the association of protein fragments with the MHC molecules differ between class 1 and class 2 molecules. How do they differ? Class 1 molecules present the degradation products from intracellular digestion, okay, endogenous proteins in the cytosol, while MHC class 2 present the fragments derived from the extracellular or exogenous proteins that are located in an intracellular component. So basically that is the difference, endogenous for class 1 and exogenous from class 2 molecules. What we know about class 1 molecules is that they are found in all nucleated cells. So proteins are fragmented, okay, by the cytosol, in the cytosol by proteasomes or enzymes. So most likely this is the lysosomes. Okay, the fragments are then transported to the endoplasmic reticulum, and then the transporter proteins and some components of the proteasome are encoded by the genes in the MHC complex. As for the class 2 pathway, so we know that there are less types of cells that are involved in that contain class 2 MHC molecules. So they are the antigen presenting cells primarily, and examples of this would be the macrophages, the DCs or the dendritic cells. For examples of that would be the Langerhans cells and the B cells. So they are also known as professional APCs, and the expression of these molecules can either be constitutive or inducible. So it's already within when it's constitutive, and inducible when it is produced artificially. So this is especially true for interferon gamma in the case of macrophages. So that would be the substance that is going to induce the um, activation of these class 2 molecules. So as we remember, this is something to do with exogenous proteins that can be taken in by endocytosis. Right, so we remember that class 2 molecules are made up of two alpha and two beta chains. So sometimes there is also the presence of an invariant chains which play an important role in antigen presentation. So this invariant chain can prevent the endogenous peptides from the cytosol from associating with the class 2 molecules, and then the class 2 molecules with the invariant chain are finally transported to the surface of the macrophages or those antigen presenting cells to the T cells. Okay, so we can see here the process. So there is the antigen uptake, the whatever antigen is that. It can be a microorganism, it can be like viruses or bacteria, and then it's taken in, uptake, and then it will be processed. Okay, and what else? There will be biosynthesis of the MHC. It will be produced. Okay, and then later on it will be associated. They will be combined together, and then later on they will be produced, or the antigen the antigen will be produced on the surface of the cell and presented to T cell. So that is for the class 2 pathway because this is exogenous. On the other hand, if it is the class 1 pathway, we know that this is endogenous. So it can be within the host itself, certain proteins are present. So these proteins are degraded, and only the peptides are allowed to be taken in by the endoplasmic reticulum. And again, it will show up in the surface of the nucleated cell presented to a T cell. So that is antigen processing by the MHC molecules. Okay, so the different professional APCs, the dendritic cells, the macrophages, and the B lymphocytes have different roles. The first role is antigen uptake. So that is the absorption of the antigen, which can happen through either endocytosis or phagocytosis. So that is also endocytosis for the B lymphocyte. Class 2 MHC expression, so the appearance of the class 2 molecules is possible because of these APCs, so the function within the cells, these cells. Another one would be co-stimulatory activity. So this can either be constitutive again or inducible. And then finally, activation of the T cell. So the different types of T cells that are activated by mm, these types of cells are the naive T cells, effector T cells, memory T cells. And on the other hand, for the macrophages, activated form of the macrophage is the one that can cause the activation of T cells, effector T cells, and memory T cells. That is also true for the resting and activated form of B lymphocytes. Notice the similarity, right? So resting B lymphocyte can stimulate the effector T cells and memory T cells like activated macrophages, and on the other hand, dendritic cells can stimulate the naive T cells, effector T cells, and memory T cells similar to activated B lymphocyte. Okay, so in this slide, we can see more like a summary of what will happen when there is the presence of organisms in the cell. So these organisms can be nucleated cells, or they can be activate antigen presenting cells. So there are two types of cells. So in the antigen presenting cells, so first there can be absorption via endocytosis or phagocytosis, and then it will be processed and digested, and it will show up in the surface of the cell being the class 2 MHC. The T cells can recognize this molecule, and it will lead to activation of the T cell. Right, so the other one is in the case of class 1 molecule. So since this is endogenous, so the substance that is already present in the cell itself is altered or changed, and then again it will show up on the surface of the molecule, MHC molecule, and it will be detected by the T cell. Take note of the difference. For class 2 molecule, the one that sees this molecule is T helper cell, while for the class 1 molecule, it's the T cytotoxic cell. And then it will be activated. These T cytotoxic cells will be activated. Okay. Now, there is a possibility that it can also interact with the products of the um, T helper cell activation. Okay, that leads to humoral immune response. So when it's already the humoral immune response, this B cell would be the main cell responsible for recognition of the antigen. So it's no longer necessary for the MHC molecules to show the antigen. Sometimes the B cell itself can readily recognize the antigen, and it can transform into a plasma cell that leads to production of antibodies. So that is the interaction between the innate response, immune response, and humoral immune response or acquired immune response. Okay, so that would be the last slide for this presentation. So we have the summary. You can check the summary of the topics that were discussed in this presentation. Right, so we have here some questions for you to guide whether or not you have already learned the important things in this slide presentation. Thank you.